What if we could literally map disease the way we map traffic, block by block, cell by cell? That's what spatial biology is doing, and it's already solving some of medicine's toughest problems. Hey, it's me, Bruno. Welcome back to Bruno's BioLogs, where we explore the coolest breakthroughs in spatial biology and why they matter. Today, we're diving into 5 real-world problems that spatial biology is helping scientists tackle right now, from cancer and Alzheimer's to developmental biology and precision medicine. Let's get into it. Tumors are never just a pile of bad cells. They're neighborhoods, each with different cell types, immune activities, and resistance levels. Spatial biology helps us map those neighborhoods, figure out who's causing trouble, and where they're hiding. In one study, researchers used xenium to look at lung adenocarcinoma and found that gene expression patterns at the edge of the tumor were totally different than from the core, and that difference was tied to how the tumor evolves. That's huge for targeting treatments more precisely. Next, neurodegeneration. Alzheimer's and ALS don't destroy the brain all at once, they spread. But where did they start and how? In one spatial study, using xenium, researchers looked at immune infiltration across layers of the hippocampus and found breakdowns in the blood-brain barrier right in the regions responsible for memory. That gives us an actual map of where the disease begins and how it might be stopped early. Now, let's talk immunotherapy. Checkpoint blockers like PD-1 have changed cancer care, but they don't work for everyone. The reason why, well, it's not just about the markers being present, it's about where those immune cells are in the tumor. In a recent study, spatial mapping of immune cells in breast cancer revealed that their locations, not just their presence, correlated with the response of PD-1 therapy. This kind of data is starting to guide patient selections in clinical trials, and it's making immunotherapy smarter. Number 4, developmental biology. How do tissues form in the first place? Well, spatial biology lets us track gene expression across space and time as tissues develop or they decide to go off course. In one fascinating paper, researchers engineered glioblastoma organoids and used spatial transcriptomics to watch the tumor evolve with remarkable similarities to the real human brain cancers. This is a big deal for understanding early development and for testing therapies in more realistic models. And finally, precision medicine. Spatial isn't just about understanding disease, it's about personalizing treatment. In a recent Cell paper, researchers used spatial profiling of biopsies in ulcerative colitis to uncover hidden inflammation patterns and define new patient subtypes. This is real-world data that's already changing how patients are stratified in trials and how drugs are chosen. So yeah, spatial biology isn't some future tech. It's here, it's powerful, and it's helping us solve cancer, neurodegeneration, immune challenges, developmental mysteries, and the future of personalized care. If something in this episode really blew your mind, hit that like button, drop a comment, and subscribe for more. And hey, hit me up, let's connect. Whether you're a student, researcher, or spatially curious, I'm always down to chat. See you in the next BioLog.